This presentation gives the details of phenotype that comes under genetics. It's an important terminology to be understood. Now here we can see definition for the phenotype and examples, and then what is phenotypic ratio with an example. You see the definition for a phenotype is observable or measurable or countable expression of every physical or physiological or biochemical characteristics or traits or variant forms of a character. What is a character? Now you say the height of an individual, whether it is animal or plant, weight of an individual, even not only the external appearance. External appearance we mean by the word observable, visible. Simply we can say how the body color is. Simply we can say tall or short, that way. But not only the visible appearance, even physiologically you can see internally what is the shape of a cell. Different types of cells are there, muscle cells, even here you see the blood cells, that way. Even the shape of cell, enzymes available in the cell, antigen produced, antibodies present, even the shape of organ among the different individuals of a population can be said as a phenotype. So accordingly, whichever the parts of the individual, if it is visible or measurable in the sense you say the height is a measurable phenotype. Countable, you say the number of RBC, a countable phenotype. So that way, a biochemical, you say the enzymes, hormones, antigen, antibody, whether even simply you can say whether they are present or absent. If they are present, you can go through the measurement, whether less, high, with this level, a higher level, that way. So all these may be called a phenotype. Now when you say the height as a character, then the different forms of the height, because in a population all the individuals may not be of the same height. Even among the human being you see some may be of 6 feet height, some may be showing 5 feet, 5.2, 5.36, that way. What are said to be the variants of a character or the traits of a character. Now if you say 6 feet as one variant of the character height, 50 kilograms as one character of one trait of the character weight. So every character may be expressed with 2 or more variants. Accordingly, the variant may be counted as a phenotype. Variant of a character may be counted as a phenotype. Now you have 100 seeds in your hand. Of them, you can count how many are round shaped, how many are irregular, the wrinkled shaped. So for the character shape of seed, you can say how many are countable, how many are round, one phenotype, how many are wrinkled, the other phenotype. If you say height in a population of individual, you can say how many are 6 feet, how many are of 5 feet, how many are 5.5. So there come 3 phenotypes, 6, the height at 5 feet, height at 6 feet, height at 5.6. So every variant may be counted as a phenotype. Now you go through the count of RBC. One may show the higher level, the other may show the anemic level. That way, the phenotype with higher level, phenotype with the anemic level. That way. So the variance may be expressed in terms of phenotype, variance of a character in a population, a countable population. Now you say, even among the Drosophila, based on the color of the eye, red eye Drosophila, white eye Drosophila. Now eye color is the character, and it is assumed that it is expressed in two forms among the individuals of Drosophila. Now you can count about 75 may show red eye color and 25 may show white eye color. So the phenotype of the eye color in two forms, expressed in two forms, physically expressed as red and white. So what is said to be the phenotype? If you are able to now for a count, for a survey, you can have a character in mind, a body color, height, weight, then see for the number of variants available among the individuals of a population. Definitely all the individuals may not show a character with the same form. The size and shape or the number may not be definitely equal, similar, what you can say, identical. So you can find the variance. So if you count the number of variants and you can name it as a phenotype of 6 feet, phenotype of 5 feet, phenotype of 5.5, phenotype of round sheet, phenotype of wrinkled sheet. So the variance may be counted in terms of phenotype. அப்போ பார்ப்பதற்கு இல்லை எண்ணுவதற்கு அல்லது அளப்பதற்கு தகுதியான ஒரு கேரக்டரினுடைய டிஃப்ரெண்ட் ஃபார்ம்ஸ், வேரியன்ட் ஃபார்ம்ஸ் ஒரு பாப்புலேஷனில் ஒரு பாப்புலேஷன் தான் எடுத்துக்கணும். யூ கேனாட் கம்பேர் த ஹைட் ஆஃப் ஒன் ஸ்பீசிஸ் வித் த ஹைட் ஆஃப் another species. இப்போது ஹியூமன் பீயிங் ஹைட்டு வேறு ஏதாவது ஒரு ஸ்பீசிஸ் ஹைட்டோட கம்பேர் பண்ணக்கூடாது. சிம்பிளி யூ கேன் compare the individuals of the same species, or if you tell it in terms of group, population. So what is said to be the phenotype? Now here I have given some examples. An observable, visible character. Simply on seeing a plant superficially, you can say whether it is tall or dwarf. And you can say there are the two variants. One phenotype is of tall type and the other phenotype is dwarf. So physically, simply by appearance, you can find whether it is tall or dwarf, distinguished as two phenotypes. Now you go with the shape of RBC among some of the individuals of human being. After processing the blood, you can see it under microscope. In some individual, you can find it is in circular shape. And in some anemic individual, very rare it may be, but anyway, possibility for the presence of a sickle shaped. You see here the appearance, the crescent shaped or arival madri appearance irukக்கூடியது, sickle shaped RBC. So now this is one phenotype. Circular shape is a phenotype, crescent shape is a phenotype, sickle shape is a phenotype. And even you go with the expression of blood group in terms of antigen, a physiological appearance, whether A antigen present, B antigen present, both A and B antigens present, or there is no antigen at all. That way you can define. Now the antigen is a phenotype, that the gene or the allele express, because you may be knowing every character is determined by the basic structures inside the cell nucleus, the gene or the allele, that we can explain it in detail. Anyway, here in what way they are expressed, that is said to be the phenotype, the external expression. External in the sense it may be inside the body or outside the body. So now here you see, you can distinguish the individuals based on the blood antigens as the individuals with AB phenotype, individuals with A phenotype, individuals with B phenotype, individuals of O phenotype, that way. Even biochemically you can measure the quantity of the enzyme, quantity of the hormone, and even the insulin level is somewhat very familiar nowadays. So that way you can define the individual into different variants, or simply for an example here I have given, if the range of the thyroid stimulating hormone in the blood of an individual to this level, they may be distinguished as of normal phenotype. And if it is less or more than this level, you can say the phenotype is abnormal. So that way. Now the phenotypic ratio may be calculated from this. Now just simply you can count the number of individuals for a particular character in different variants. Now here you see the example. If you count for the number of testers and non-testers regarding the PTC, phenyl thiocarbamide, the substance, if you put a drop on the tongue, some of the individuals only can feel some taste, a bitter or sour taste, and some of the individuals may not find any taste out of that solution. Accordingly, you can define the taster phenotype, non-taster phenotype. And among the individuals of 100, if you are able to find taster 75, non-tasters 25, you minimize the number to the possible extent. Now here you see the 75, 25 reciprocally minimized. You can tell it in terms of 3:1 earlier. If you write it in the form of ratio 3:1, that may be the phenotypic ratio of the character ability to taste. So here you see the actual number of variant of a character surveyed and reciprocally minimized and written in the form of ratio. What is said to be phenotypic ratio.